So this session is on big ideas, and I want to present a new perspective on an old idea, and that is how we identify the genetic regulators of cell state. So we can look at this in the context of development. What are the genes that govern pluripotency? Sorry. All right. Well, anyway, what are the genes that are governing pluripotency? What are the genes that govern definitive cell types like cardiomyocytes or endothelial cells? Thanks. And what are the genes that govern the cell transitions through developmental processes? We can look at this also in the context of disease. If a tissue is injured, what are the genes that are governing the response of a cell to that change in the environment? These are fundamental questions about developmental biology and the etiology of disease. Anyway, we have a number of tools at our disposal these days to be able to address these questions. We can look at the nuclear architecture, the interactions within the genome. We can look at the proteins bound across the genome using DNase hypersensitivity. We can look at transcription factor binding or histone modifications that allow us to elucidate specific functions of different parts of the genome. And we can look at the transcriptional profile of a tissue or a cell type. And these give us different pieces of information about what's going on at the level of the nucleus that allow us to be able to dissect some of the mechanisms that are governing cell identity and cell state. Now, I'll say that historically, I'll just make a conjecture here, but I think that there has been, either because of limitations in technology or whatever the case may be, there's been a significant emphasis on transcriptional profiling if you think of quantitative RT-PCR or the array approaches that we had in the past or contemporary approaches in RNA sequencing. The effort to understand the function and identity of a cell has predominantly been focused on understanding the transcriptome. And we've taken, an old approach here that I would say, maybe older, getting older, is to take a population of cells through development or to compare two different cell types and getting this average signal that you might say we have a novel gene that we're interested in and how does it compare to the transcriptional profile of other known genes. And then we can use immunohistochemistry or FACS analysis to look at the single cell level, more specific mechanisms that are related to the expression of that particular gene of interest. But this has gotten us into a new phase of our understanding of transcriptional profiling at the single cell level. So now, instead of looking at populations of cells and taking the mean or the average signal of a particular gene of interest, we can now look at the transcriptome of every single cell, where we're looking at endothelial development here, for example, where we have nascent mesoderm transitioning through to endothelium and the blood lineage is here. And every single cell basically acts as an entity in itself, where we can look at the transcriptional profile of every one of those cells and then look at a gene of interest, such as HOPX, for example, and see its distribution within those populations. And this is fantastic. It gives us an excellent opportunity to be able to get at mechanistic insights at a single cell level. Anyway, so these are really excellent technological advances that we have at our disposal these days. But let me just say that there are some significant limitations to transcriptional profiling that we're really running up against some roadblocks in this. And that is that it is only profiling the most highly abundant transcripts. And that's a problem because transcription factors, for example, or other key regulators of a cell's identity that are governing that identity are in very low abundance. And so you're missing out on a great deal of information simply because you can't actually detect it. On the other hand, we're also looking at perhaps differential expression. This is a classic approach where you're looking at two populations of cells and looking at differential expression. And again, this is limited on the abundance. You pull this twofold differential expression, and that's some arbitrary line that says that those that are in highest abundance are somehow more functionally important. But I would say, anyway, what I'm trying to get at here today is that this is actually a major limitation, that we are not actually able to elucidate the fundamental genes that are governing the identity of a cell using transcriptional profiling alone. So I want to back up a little bit here and focus in on some of our newer approaches here, and that is specifically focusing in on ChIP-seq and chromatin dynamics. And so we have an opportunity to be able to identify and analyze the entire genome, looking at enhancers, active or primed enhancers, super enhancers, open chromatin, closed chromatin. The list goes on. We have these histone tails that are modified in specific ways that allow us to be able to dissect out specific regions of the genome that allow us then to get at some more mechanism. And I've taken an approach in recent years to try and incorporate chromatin dynamics in a manner that allows us to be able to get more specifically at the genes governing cell identity. So if you have these reads, for example, we'll have a number of different populations of interest over here. We have a number of chromatin marks here as it lines up with RNA sequencing profiling here. And the question that is at hand here that I want to ask is, how do you tell the difference between a structure function gene here, like troponin I, or a transcription factor like NKX2.5 or tau1? How do you actually get at identifying these specific genes, specifically new regulators of cell identity or cell fate, using chromatin dynamics in combination with gene expression? And so I'm not going to go into this. It's very complicated, and honestly, it's very laborious algorithm. But the basic essence of what we've done here is we've taken two approaches. On the one hand, we take our traditional approach of just taking transcriptional profiling. We have a couple of populations of interest, and we look at differential expression. So that's panel number one. That's our one approach. The other approach then is to say, we're going to incorporate the transcriptional profiling in combination with chromatin dynamics to see if we can more specifically enrich for the genes that are governing cell identity. And so if we look at the genes that are enriched simply on the basis of gene expression, and this is looking at the cardiac lineage because that's the lineage I'm most interested in, you'll see that by looking at just gene expression differences, we're picking up sarcomere organization and muscle contraction, structural constituents of muscle. And these are the genes that are in high abundance, the transcripts that are in high abundance, like myosin or troponin. These are just functional elements of a cardiomyocyte. But those are not the genes that are governing the identity of that cell. So when we incorporate the chromatin dynamics, the profiles change here. We now are looking at developmental processes, morphogenesis, cell fate commitment, DNA binding. So now we've whittled away all of these high abundance transcripts, and by incorporating chromatin dynamics, now we have a more specifically enriched population of genes that we think are governing the identity of that cell. So let me give you an example of one gene that we picked up using this analysis. And I highlight this particular example because it's not a transcription factor. We have sort of this unreasonable idea that transcription factors are the genes that are governing cell identity. But the cell has a very different interpretation of what that means. And the chromatin dynamics actually create a particular signature of genes governing cell identity, like transcription factors, but not specifically transcription factors. And this is a transmembrane protein that has chromatin dynamics that recapitulate exactly what you might see with a transcription factor. So let me walk you through this. If we have here pluripotency, and the top is H3K27 trimethylation, which is a closed repressed chromatin, H3K4 trimethylation, which is open chromatin, and then H3K36 trimethylation, which is a surrogate for RNA polymerase activity. So in the pluripotent state, this gene, TMEM88, is heavily repressed. And as the cells transition through mesoderm, you say that the repressive mark is still present. And then as the cells transit toward the cardiac progenitor cell, this is replaced with open chromatin marks. So now the repressive brakes are released, and the gene is now actively expressed. And you can see that as it transits on toward a more definitive cardiomyocyte, anyway, that signature dies out. But we carried out functional assays on this, and we found that when we knock down TMEM88, we lose the ability to be able to make cardiomyocytes. This can be rescued with a Wnt inhibitor, which is in keeping with the fact that TMEM88 is an inhibitor of Wnt signaling. And the cells transited instead into an endothelial fate based on this immunohistochemistry for von Willebrand factor. So the key point that I want to make here is that the ability to draw in chromatin dynamics enabled us to identify this novel transmembrane protein that is critical for being able to identify and mechanisms underlying cell fate commitment in cardiovascular development. So in closing, I just want to reiterate my key point here, and that is that transcriptional profiling is good. It's fantastic. It's important. Single-cell RNA sequencing is really amazing, and there are undoubtedly going to be fantastic discoveries made through this, but there are limitations to transcriptional profiling that in terms of being able to get at the key genes that are governing cell identity. And I think that we need to draw our focus and attention toward chromatin dynamics as an approach, a unique opportunity to be able to take the cell's interpretation of key genes that are governing cell identity at the level of histone modifications and this relationship between open and closed chromatin as it relates to gene expression. So with that, I will close. I have a small lab right now up in Brisbane, 2 folks growing. As well as some fantastic collaborators from the United States where I came from last year, and funding sources for my work. So anyway, thank you for your attention, and I think we'll have questions at our panel discussion. Thank you very much.